this video, we're going to be looking at some past paper questions based on the content of our Genetics 2 video. So from November 2019 in paper 2, this is from section A. The diagram below shows the pattern of inheritance of a disorder. Don't be put off by the fact that it says male-female. Don't assume that this is a sex-linked disorder. In fact, it isn't. So if the male is not affected, he will be capital B, and we're not sure of his second allele. If the female is not affected, she will be capital B. We're not sure of her second allele. We've just chosen B's at random here. The child, however, is affected, and so the child will be small b, small b. That means that both parents must be heterozygous. Okay, knowing that, now let's look at the options. One can conclude that the disorder is caused by a recessive allele with both parents heterozygous. That is correct. Let's just check why it can't be the others. Is it caused by a dominant allele? No, because if it was caused by a dominant allele, both the father and the mother would have the disorder, because if a dominant allele is present, it will be expressed in the phenotype. Could it be C, a recessive allele with one parent homozygous recessive while the other is heterozygous? That is a possibility, except that if one parent was homozygous recessive, they would be little b, and neither of the parents is affected. D, a dominant allele with one parent heterozygous and the other homozygous recessive. Again, it cannot be that, because if a dominant allele causes this disorder, then both parents would have the disorder. If the dominant caused the disorder, it would show up in the phenotype. All right, here we've got a question looking at mutations. So you've got a piece of DNA before, a mutation, and a piece of DNA afterwards. So will this mutation result in an extra chromosome? Will it produce the same protein if a different amino acid is coded for? Will it produce a different protein if a different amino acid is coded for? Or is it the result of an extra chromosome? So let's have a look. The difference in the DNA is that base. AACTA GCTT becomes AACTC GCTT. Now remember that we read our DNA in codons. So let's put in our codons. So what we're looking at here is, will TAG code for the same amino acid as TCG? If it codes for a different amino acid, then it cannot code for the same protein. If it codes for a different amino acid, then it does code for a different protein. Neither A nor D is correct, because we're looking at a portion of DNA, not a whole chromosome. And so C is the correct answer. Which one of the following best describes the mutation? More than one nitrogenous base was changed. Adenine was changed to cytosine, adenine was changed to thymine, or cytosine was changed to adenine. Adenine was changed to cytosine. You're told a certain marine invertebrate, the color of the shell is under the control of one gene with 3 alleles. In different combinations, the 3 alleles produce 4 phenotypes: orange, yellow, and black. Orange-yellow, being a hyphenated word, means that is incomplete dominance. The table below shows the results of the offspring produced from crosses involving parents of different phenotypes. The first question says, name and describe the type of dominance shown by cross 4. You can see in cross 4 that it is orange crossed with yellow, which gives us all yellow. Orange-yellow, which, as we've said before, because it's a hyphenated word, means that is incomplete dominance. Now it says describe. Notice it's for 3 marks. So simply naming it gives you 1 mark, but now you need to describe this dominance. And the way you would describe it is to say, both alleles orange and yellow are dominant, and when present as a heterozygous combination in the offspring, they produce an intermediate phenotype, or a blended phenotype, orange-yellow. That would get you your other 2 marks. Which shell color is controlled by a recessive allele? Now, the fact that you've got black indicated in the first paragraph, and that they're telling you that there is now a recessive, tells you that black must be the recessive allele. If orange and yellow are incompletely dominant with each other, producing orange-yellow as our blended phenotype, black must be the recessive. And then it says, use information in the table to support your answer. Well, if we have a look at cross number 1, there we have yellow crossed with yellow, which produces some black offspring. And in cross 3, we have orange crossed with orange, which also produces some black offspring. Therefore, black is recessive to yellow, and black is recessive to orange. A mother has blood group B and a father has blood group O. They have 3 biological children and an adopted child. The blood groups of all 3 children are represented in the table below. Which child is adopted? All right. If the mother is blood group B, she could be IB, or she could be IB little I. The father is blood group O. So we need to do 2 quick Punnett squares to determine which one of those is going to be able to give us the combination of phenotypes that we see in the table. So if the mother is IB and the father is little I, then all of the offspring are going to be blood group B. That's not what we see. So the mother must therefore be IB little I with the blood group O father, so if you look at that table, you can see that the offspring here would either be blood group B or they'd be blood group O, which means that Millie with blood group AB must be the adopted child. Here we have an item statement question. The type of inheritance where both alleles are expressed equally in the phenotype, is it A only, codominance, B only, complete dominance? Is it both A and B, or is it neither A nor B? Remember that in the exam, you must write A only, you must write A only, B only, both A and B, or none. Those are the words that you must put in your book. So we're looking at alleles expressed equally in the phenotype. If they are expressed equally, it is codominance. In the exam, you are definitely going to get some kind of case study looking at some aspect of genetics. The case study that you get will always, or almost certainly always, be on a particular disorder or disease that you have not come across before, or that it is highly unlikely that you will ever have come across before. Don't panic when you see something that you're not familiar with. Read through the question carefully, because the questions themselves will be things that you can answer. So this disorder, SCID, affects the immune system, and it is caused by a sex-linked recessive allele. So the fact that they've given you that code X with the D in it tells you that you need to write that special notation. The diagram below shows the inheritance of the disorder in a family. It is not known if individual 1 has the disorder or not. The fact that they've pointed this out to you should give you a clue that there's probably going to be a question in which you have to do a test cross and work backwards to work out what the genotype or the phenotype of individual 1 is. So it says, give the phenotype of individual 2. In the key, individual 2 is a female without SCID. Now remember, phenotype doesn't mean she's not heterozygous. It means that she appears normal. So phenotype of individual 2 is a female without SCID. Give the phenotype of individual 1. So now we have to work backwards. Individual 4, as you can see from the key, is a female with SCID. So she's going to be X little d, X little d. The mother, as we've just seen, is normal in appearance. So X capital D, X, and we're not sure what that one is. Having said that, in order for individual 4 to inherit 2 copies of the recessive allele, she has to get 1 from her mother, and therefore this second allele must be the recessive allele. This first allele she must therefore get from her father. Now, the fact that we are looking at a sex-linked disorder means that the male only needs 1 recessive copy to have the disorder. He is going to be X little d, Y. So the phenotype of individual 1 is going to be a male with SCID. What is the genotype of individual 3? Well, individual 3 is a female, so it's XX. We know that there is a possibility that she could inherit the recessive allele from her mother. However, if she did, she would be affected just like her sister, individual number 4. She is normal in appearance, so she must actually have inherited the dominant gene from her mother. And that means the only X she can inherit from her father is the recessive allele. And so individual 3 is X small d. 2.2.2 says explain how individual 5 inherited the disorder. Well, we know what the genotype is of the mother. We know that the father must be normal because he only has 1 X chromosome. And so if he had a small d, he would be affected. The Y chromosome from the father has come to individual 5, which means that 1 of the recessive alleles must come from the mother because that's all the mother can give, and so he must be X small d, Y. So now, explain how individual 5 inherited the disorder. So you would say, because he's male, he gets his Y chromosome from his father, and his X chromosome from his mother. So that together would give you 1 mark. Then you would say his mother's genotype is X little d, meaning that she can only give gametes with an X little d chromosome or allele. And that would get you your 2nd mark. 2.3 is a question about cloning. So we have the first cloned animal in Africa, the calf named Futi. No fertilization involved in her production. She was produced from a single cell taken from the ear of donor cow LMJC 865. LMJC 865 has a high average milk yield. She's able to produce 78 liters of milk a day. And then there's some basic other information. So according to the extract notice, state one advantage and one disadvantage of cloning. So you could literally state that particular sentence: cloning allows for the production of organisms with desired characteristics, as your advantage. And as your disadvantage, you can state that sentence: cloning reduces genetic variation in the offspring. You could also have said, for your disadvantage, that it is an expensive process. It may not be economical for commercial agriculture. State why the donor cell was taken from LMJC865 and not from any other cow. Well, quite simply, she's a very special cow. She can produce 78 liters of milk a day. Why did we use an ear cell and not an ovum? Because what we're looking for from the ear cell is the nucleus, which has all of the genes. We do not want to dilute those genes by going through meiosis and then adding in a second set of DNA from another individual. Briefly describe the process of cloning. Notice that it is only worth 4 marks, so it literally is a very short paragraph. So you would say something like, the nucleus is removed from a donor cell or a donor body cell. It is inserted into an empty or a vacant egg cell. After electricity is applied, that cell begins dividing like a zygote. It is then implanted into the uterus of another individual, where the zygote continues to grow and gestation continues as normal. Moving on to November 2017. A very straightforward question here, 112. What is the genotype of an individual with blood group A? So blood group A, you can either be homozygous dominant or you could be heterozygous. So D is the correct answer. Color blindness, which is also known as daltonism, is a sex-linked disorder caused by a recessive allele X(d). Again, notice that they say it's a sex-linked disorder. That means that when you write the information down in your answer, you must use the special notation. So there is our table of this family. Give the phenotype of Senzo. Using your key, you can see that Senzo is a colorblind male. Give the genotype for Tuli. Using the key, you can see that Tuli is a normal female, but they've asked for genotype. The fact that we have Linda, who is a female who is affected, means that she must have inherited 2 X chromosomes that both have recessive alleles, one from her mother and one from her father. So her mother, therefore, must have the recessive allele, but her mother appears normal, so she must be a heterozygous individual. Describe how Linda inherited her colorblindness. Well, we can see that Linda is a colorblind female, so she must be X(d), X(d). She gets 1 X(d) from her mother. Her father, who is colorblind, gives her the other X(d). So you would say, because it's only worth 2 marks, you would say she inherits 1 recessive allele from each of her parents, or 1 from mom and 1 from dad. Explain why there are generally more males than females with colorblindness in a population. This is for 4 marks. They're asking you to explain how or what the purpose is of the second X, essentially. So you would say, males only have 1 X chromosome, females have 2. If females inherit a recessive allele, they usually have a second allele that could be expressed instead, which is more likely to be a dominant. If a male inherits a recessive allele on his X chromosome, he does not have a second copy of that chromosome and therefore does not have an alternative allele which could be expressed. This means that males in general tend to experience more sex-linked disorders like colorblindness than females do. Molly married a normal male. Use a genetic cross to show the possible genotypes and phenotypes of their children. Okay, this is a monohybrid cross question. You're going to write out the whole template. Molly is a normal female. However, because her father is affected, she will inherit an X chromosome from him, which carries the recessive allele. Molly is therefore X big D, X little d. So now, let's do this monohybrid cross template. Now we're going to look at the F1 genotype. And the F1 genotype is going to be 1 to 1 to 1 to 1. You're going to have your homozygous dominant female, your heterozygous female, your dominant male, and recessive male. Now, if we look at the phenotype, remember that if you have one dominant gene, you're going to be a normal individual. So these two are normal, so you're going to have let's rather put that as a percentage. You're going to have 50% normal females. You're going to have 25% as a normal male, and 25% will be a colorblind male. All right, let's move on to 2016. Now, because genetics and DNA are so closely intertwined, you will often find in one question they ask some DNA questions and some genetics questions. So here's a question looking at DNA profiling. It says there's this picture based on a particular procedure. What is the procedure? The procedure is DNA profiling. It then says to you, the evidence in the diagram shows that which individual was present at the scene. You can see that the DNA bars, you can see that the DNA bars match up quite nicely with Joe. So Joe is the one who was present at the scene of the crime. However, if you look further to the right, you will see that those bars also correspond with Tom. So both Joe and Tom were present at the scene of the crime. And here comes the genetics question. Below is a list of the possible uses of the procedure shown in the diagram above. Paternity testing, matching of tissues, identification from fingerprints, screening for genetic disorders. Which combination shows the correct use of the procedure? Don't be confused or don't be misled by the use of the word fingerprints. Number 3 is not a possible procedure. That means that only 1, 2, and 4 are possible uses. And 1, 2, and 4 is answer B. All right, question 2.4. Here is another disorder that you may not have come across. And again, it's a case study looking at a pedigree diagram. So Huntington's chorea is a disease caused by a gene mutation that results in the degeneration of brain tissue. It's caused by a dominant allele, H. The fact that H is written just like that and it's not X, capital H, means that this is not a sex-linked disorder. This is an ordinary monohybrid cross question. So there we've got our family tree, our pedigree diagram, showing the different individuals who have inherited Huntington's. What is Susan's phenotype? Susan's phenotype, because she's colored in, she is a woman with Huntington's chorea. So we're going to say what is Susan's phenotype? Because her circle is completely colored in, we can read from the key that she is a woman with Huntington's chorea. What is Susan's phenotype? Because Susan's circle is fully colored in, this means that she is a woman with Huntington's chorea. What is Sarah's genotype? As we've seen in a previous example, Susan, if she is, as we've seen in a previous example, Susan is little h, little h. One of those little h's will come from her father, and the other comes from her mother, which means that Sarah's genotype must be heterozygous, because Sarah's circle indicates that she does not have Huntington's chorea. So Sarah's genotype is big H, little h. Emma plans to have a baby. What must the father's genotype be so that there is a 50% chance that their child will not have Huntington's chorea? Now if we do a little Punnett square, we know that Emma is little h, because she already has Huntington's chorea. In order for 50% of the children, or 50% chance for the children, so those two, her partner must have one capital H. However, if he has another capital H, none of their children run the risk of inheriting the disorder. That's not what the question asks, though. The question asks, what is the chance that only 50% of them will not get the disease? And that is the 50% that will not get the disease. So therefore, her partner must be capital H, small h. And then for 3 marks, it says explain your answer. So exactly as I've gone through now, you would do 2 Punnett squares, one with the potential partner being homozygous dominant, and one, as it shows there, with the partner being heterozygous. Once having drawn your 2 Punnett squares, you would then write a concluding statement saying, only in the one Punnett square is there a 50% chance, and therefore the father has to be big H, small h. You have 2 rose plants, both with pink flowers, and you cross them to find that while most offspring are pink, some are red and some are white. Okay, the fact that there's pink, this immediately tells you that we are looking at a blended phenotype, and so incomplete dominance. Use a genetic cross to show how breeding 2 pink flowers can result in pink, red, and white flowering plants. Use the letter R for red and W for white. F1 genotype. we have a 1 to 2 to 1, RR to RW to WW. And the phenotype will be 1 to 2 to 1, because remember, the heterozygous condition in this case is not going to display the dominant characteristic. And so that is red to pink to white. And our last question, looking at genetic modification. So genetic modification began with the discovery that the soil bacterium, Agrobacterium, could be used to transfer useful genes from unrelated species into plants. The gene, called Bt, which produces a pesticide toxin that is harmless to humans, but is capable of killing insect pests, is one of the genes most commonly inserted into crop plants. Many new GM crops like maize, potatoes, and tomatoes are modified to be pest, disease, or weed killer resistant. GM foods could have unforeseen effects. Toxic proteins may be produced, or antibiotic-resistant genes may be transferred to human gut bacteria. Modified crops could become weed killer-resistant superweeds. Modified crops could also accidentally breed with wild plants or other crops. So according to the passage, how did genetic modification of crops begin? It was with the discovery of Agrobacterium, which can be used to transfer useful genes from unrelated species into plants. Remember that when you write Agrobacterium, because that is the scientific name of an organism, the A must be a capital A and the entire word must be underlined. Explain why a plant which is modified to be weed killer resistant could be a problem for farmers. So you would explain that the potential of accidental breeding between the crops and, for example, weed species could result in weeds that are weed killer resistant, which means that the farmer would then have to increase his labor to be able to get rid of the weeds and to save his crop. Give 2 examples in the passage of the use of GM crops that may be a potential threat to human life. Directly from the text, we can take about antibiotic-resistant genes that can be transferred to human gut bacteria. And if those bacteria are harmful bacteria, we would then be permanently sick with vomiting or diarrhea or something like that. And just above that, toxic proteins may be produced. So you might eat something that has a toxic protein in it that could be very harmful to you and give you poisoning.